Hello, and welcome to our webinar, Advanced Analytics for Batch Process Improvement with Grantek and Seeq. Thank you for joining us. A few things to mention before we begin. Please submit your questions through the questions section of the GoToWebinar console. If we do not answer your question during the session, we will follow up with you afterwards. For any additional questions or inquiries, please email us at info@grantek.com. Seeq resources can be found on Seeq.com and on our community site, Seeq.org. Our presenters for today's webinar are Bryon Hayes, Director of Life Sciences at Grantek, and Joseph Reckamp, Analytics Engineer at Seeq. And with that, I will hand it over to Bryon.
Thank you, Elise. I'm very excited to be here. So thanks to you and to Seeq for having me. To give a very brief background on Grantek. We're a systems integrator. So we're a small company, but we're considered a large integrator within North America. We have offices located across the US and Canada, as well as in India, and we pride ourselves in being a complete business solutions provider. What I mean by complete business solutions provider is that we strive to see beyond the implementation of projects and the solving of technical challenges in manufacturing, instead focusing on the wider implications of the work we do, specifically how it affects, improves, and enables our customers' business. One of our primary goals is to lead our customers through transformative business and manufacturing initiatives, using our experience and knowledge to guide them toward becoming best-in-class practitioners of smart manufacturing. As a matter of fact, one unique thing about Grantek is that our heritage is in engineering, but we also have expertise in navigating the complex compliance landscape inherent in highly regulated industries, such as pharma and life sciences. We are able to share both our technical and our industry experience and knowledge with customers in the space, which in turn drives successful business outcomes for them. Many of our core competencies have their roots within the four walls of the manufacturing plant, where we are engaged in process automation, industrial networking, packaging systems, and the like. But we've really grown as a company in extending our focus more toward interconnectivity and Industry 4.0 solutions, including connecting our customers' plant floor equipment with their business systems and extending the reach of manufacturing into the cloud to enable new technology innovations that all drive value back to the business. Specifically in the pharma life sciences landscape, we've been asked by our customers to help them leverage their massive volumes of data to derive insights that enable them to make informed business decisions and support initiatives such as continued process verification. This is where Seeq has been an important enabler and partner for Grantek. And with that, I'll turn it over to Joe to describe what Seeq can do for our customers.
Yeah, so let's first start off with talking a little bit about what type of analytics there are out there. So when you think about the different types of analytics available, there's kind of 5 different approaches. There's ones that first focus on the past, things like diagnostic and descriptive analytics, where you're looking at, what happened or why did something happen, what went bump in the night that you might want to look at that root cause analysis of what might be happening there. You then have the current or near real-time things of what's actually happening currently, the monitoring aspect of what's going on in the current batch as it's running right now. And then you have things to try and help you enable, give you that opportunity to improve the future outcomes. So things like predictive analytics to describe where are things going in the batch or in the process overall, and how can we predict what might happen, or how might we prescribe different changes to avoid a certain scenario or improve a particular outcome in the batch. So Seeq covers all of these, and it starts off with Seeq Workbench. And Seeq Workbench is really the workhorse of the Seeq application. It's a browser-based application, so just in your favorite modern browser, gives you access to all those data sources that you might connect to, and we'll talk about that in a little bit, with various tools to enable things from analytics, calculations, visualizations that might be there, even data cleansing or pattern recognition that you might want. Once you have that valuable outcome that you might care about, we can then go into Seeq Organizer, where we can share and collaborate with others to report that information. So Seeq Organizer is our dashboard or reporting application to make those automated workflows, to continue to track your batches as they come out, even doing things like annual product reports or things like that. And then finally, Seeq also has a Seeq Data Lab component, which is for really the data scientists. So people that are interested in operationalizing machine learning models, interacting with the Python environment. We have a SPY module, the Seeq Python library, that allows you to access that data and perform any of those calculations or advanced machine learning applications on it, and push that data back into Seeq to really interact and collaborate with not only just on the data science side, but also with the subject matter expertise that understands that data. All three of these applications live kind of above and connected to this Seeq server. And this Seeq server is the calculation engine behind everything. It also includes all of the data source connections. So you may have a variety of different manufacturing data sources. So historians like OSIsoft or GE Prophecy. We have native connectors to all these various historians and many more that aren't even shown here, along with SQL-based data systems. So we often find that customers have LIMS data or R&D laboratory data in these SQL-based systems. So we have connectors to those as well. And then finally, there's a wide variety of custom sources, from forecast weather data to specific lab instrument data. We have an ability to kind of connect to that data and bring it in. And those connections bring that data to that Seeq server and allow all these other applications to access it. So whether you're running a machine learning model on that data, or you're just reporting out a trend that might be seeing in the data, all of these Workbench, Organizer, and Data Lab all connect to that Seeq server. We also have different integration options. So you may be using BI tools like Tableau, Spotfire, or Power BI. You might have pre-written code that you might have written in MATLAB or something that we can connect to with an SDK there. All of those are different integration options that are available within Seeq. So when you think about the current landscape of what's out there in process manufacturing, particularly in life sciences, there's a lot of data out there. There's sensors all over the plant that are collecting tons of data and storing that in your various historians and databases that are out there. What we find is that there's actually a lack of use of that data. There's less information being gathered from it than could be. And this is because people are trying to do manual analyses in spreadsheets where you have to, do an analysis every batch and recreate it every time you want to redo that. And it leads to kind of long time to insight and an inability to find that in a reasonable manner, and these inflexible or static reports that you might see. We found with working with customers that it's not just good enough to put in a technology and see what it can do. You actually have to define that scope. So what is that problem that you actually want to solve, and what is that return on investment that you can expect to get the approval of putting in these sorts of technologies and financial justification of that. And so what we want to challenge everyone on the call to do is actually really understand what are your use cases and what problems are you trying to solve, and then really evaluate and pick the right solution to solve that. One thing that we see with pharmaceutical customers in particular, as well as other life sciences customers, is that there's this aspect of kind of a continuum from the lab R&D scale all the way up to commercial, where in the lab, they spend lots of years and many different experiments testing out and designing that process. And in that time frame, when you're looking at that lab data, you're creating all this data that could be used at the pilot and commercial scale, but really isn't translated or available to those operators when you get to the commercial application. And so what we found with Seeq is that customers have started to document all that information, the quality by design aspects of it, whatever's going into those lab experiments and the defining of the process, capturing it into Seeq and actually transferring that across to the live connections to pilot or commercial data, so that you can really understand those relationships and what parameters are affecting your process models in the lab scale. You can kind of build on that and actually verify the principles of scalability and things when you go into the pilot scale, and then actually have those golden profiles or prediction models or whatever it might be available automatically at the commercial scale as operators try and strive for the best possible batches. This allows, when there is a deviation that occurs, they can go back and see it. Has this ever happened before? Was this found in the pilot batches or the lab batches that happened previously? And can we see what sort of outcome or what sort of response there might be that would address that? So let's start talking about some actual use cases here. So in this first use case, we're going to talk about clean in place technology, so CIP cycles. And so what we find is that cleaning systems are often based on a timer. So they've got a set amount of time that they're going to spend in each phase of that cleaning operation. And they really aren't using the data to drive when the cleaning cycle is actually complete. And there might be some optimization of, saving time, saving CIP solution that could be achieved there. And so kind of the solution within Seeq was actually to, determine when is the system actually clean. So when is that desired CPP, whether it's a conductivity value, a UV value, whatever value is determining what aspect of this product is gone from the system. Let's find out when we actually achieve that, compare it to that actual cycle time that we're operating under currently, and create KPIs to actually evaluate how much savings potential there are out there. And so ultimately, this allows you to, reduce time and resources used in that cleaning process, but also leverage those KPIs to actually identify, how much time and savings are we actually creating with this aspect of data-driven approach to CIP cycles. So let's go in and see this actually in action here. So we're going to start off by quantifying in Seeq by opening the Seeq overview screen, and we're going to go into a workbench analysis, so that Seeq workbench that we talked about earlier. So first, we want to showcase a journal feature to illustrate how we built out this use case. The journal itself is an effective way for the experts to collaborate and share their thought processes with their peers. This is a great way to capture information from the tech transfer process, etc. Journals can contain text, as you kind of see here, or they can even contain links where you might have links to tags or views or time ranges, calculations, etc. that might be in there. To display this journal in action, I'm going to go and actually add some tags to the screen. So we're going to go over into our data connections and find some of those tags that might be relevant for this, just to kind of showcase this feature. So in this asset tree here, I'm going to go grab just a few tags on the screen, create a custom condition and just find a period of time that might be interesting, and show you kind of how you can go in here and hit the edit journal entry and add information, just some text that you might want to add a link to or something like that. We can highlight this text, and using this green button in the corner for the insert a Seeq link, we can add links to either the entire work step, the specific trend items, or time ranges that are on the worksheet, or even individual capsules, which might be individual batches or something. So generally, we use the current work step the majority of the time, which saves the entire screen. So all the signals that are on the screen, the time frame of interest, etc. Just to kind of show you this in action, we're going to remove everything on the screen, kind of mess up the display of sorts. Then you can see when you go back and click on this link here, it's going to bring everything right back to where we were, so that you can really quickly get through that analysis as to what you were doing in your thought process as you were building out those analytics. So before we get going, let's remove that example link and then go back to the actual CIP cycle. So in this cycle, we're optimizing some CPPs for clean in place. And so we're going to first start off with the CIP phases themselves. So these might be event frames in an OSIsoft Pi database, or it might be SQL database or MES system that you might have these CIP phases defined. In this case, you can see in the bottom right corner that we have those phases of pre-rinse, wash, and final rinse that we're kind of working through. And we want to evaluate the conductivity and flow variables along with that. So we're going to add those to the screen by clicking on the next journal link. Now, when you look at this, it kind of relates to the same thing where we've got the time frames where the flow is actually present. And when that flow is, actually above zero, you can see those pretty well align with any of those CIP phases. We can also find the times when the conductivity is low. So below a certain limit that we set, maybe 400 microsiemens per centimeter. And so this is done with a quick value search, which is in our tools panel inside Seeq. You can see it gives you that capsule at the top of the screen in yellow. Now, what we're really interested in is that time that I've grabbed there, which is the overlap. When were we actually clean, but we were still running our CIP solution? That's our potential savings that we might have. And so let's identify that automatically by using a composite condition in Seeq, which can give you the combination of when those two, the pink and the yellow, intersect there. And you can see it grabbed that with that new purple capsule there. Now, from there, we want to actually identify some of the KPIs. So things that you might think about are, how much time would we have saved if we would have ended the cycle earlier when that clean limit was actually reached, or when how much utility savings of the CIP solution we'd actually have. And so just as an example, we have the utility savings here, basically integrating over the course of time, the total flow rate signal there. And you can see that there's approximately a little under 50,000 kilograms at the end of the cycle that we've wasted. We can also view this in a different way in terms of a scorecard where we have a tabular view, which is calculating over that time frame, how much savings do we have in utilities as well as total minutes. And we can turn this over into an organizer topic if we want to share it with the operators, set this up to an automatically updating report, kind of have the trends with the scorecard view all together in one spot. And we've had customers that have used this sort of system to save millions of dollars per year by reducing those raw material waste and optimizing these batch production cycles. So this is just an interesting way to add that context to the data and understand and quantify and be able to really track what's happening in your process over time. Moving on to the next use case here, we have a batch quality prediction example. So one thing that we often find is that there are lab results that are taken. So in a batch process, you often take a sample of the reactor and send it off to the lab and either wait for a response or keep moving on with the batch while you try and figure out if your quality actually meets its metrics. And so what this makes it is it's a delayed response to all your process variables. So instead of being able to optimize what's going into the process and kind of optimizing that yield or quality in real time, we actually have a delayed response because we're actually waiting for a quality result. And at that time, it's too late to affect that parameter. And so what we want to do is actually create an online model. In this case, we're going to do it for yield, but you can also do the same thing for quality based on concentration, volume, temperature, whatever parameters in your system that you might want, and have that operating basically near real time as the data is coming into the historian. This kind of results in these, rapid identification and root cause analysis of abnormal batches, can save energy, save batches, save, problems with quality, et cetera. And, building these process models, allowing this historical information to predict product quality or process outcomes is much better than waiting for that offline result. These multivariate models, even in the R&D phase, can allow you to really impact which parameters are critical to the process and understand that so that when we deploy it online, we know which parameters we can actually influence as we're kind of building out the process. So let's kind of see this prediction modeling in Seeq. So in this case, we're going to start off and we're going to add the batches and the process variables to the screen first. And so you can see here we have 3 different process variables: level, temperature, and concentration, along with kind of a Gantt chart view of specific batches at the top of the screen. And so what we're doing is filtering out the batches to a particular product. So in this case, we're using just the batches that display the green product. And we're then filtering those even further to say, we just want the operation when a particular processing step is running in this case. So we'll ignore the cleaning, the idle times, et cetera, and we'll just grab that particular processing step to work off of. And so we can view this data in chain view, so we can kind of line up just those processing steps next to each other. And you can see kind of the similarity between all these different steps as you go through. And then the final parameter to add to the screen is the yield value. So after the fact, we've got these lab results coming back with % yield, and we've lined these up with the batches to identify, what is that percentage yield as it comes out each time. And what we want to do is use these historical batches to actually predict what parameters influence that yield value. And you can take this by, looking at the parameters. That if you look at the number of counts of samples of these, we have over 10,000 samples of these level, temperature, and concentration, but only 10 yield values. So we need to find a way to model this single value per batch as a function of the overall trajectory of this batch as it goes forward. And so one way to do this is to use engineering principles and determine, what statistics might apply to these parameters that we can actually use based on our process knowledge. So for instance, maybe the maximum temperature that we achieve is an important parameter. So we can calculate out using our signal from condition tool, the maximum temperature per batch and say, every time we run this phase, let's find what that max temperature was during there. And let's plot that so that we can use that as a single variable per batch as the input to those models. In addition, we've done the same for the maximum concentration and the standard deviation for the level. And you can see in this case now we have 10 individual values, one for each batch, that we can easily model appropriately. And so the next step from here is to actually kind of look at this as data and verify that we're set up and we're clean, we're ready to go to build this prediction model. And so what we'll do now is add that model to the screen using our prediction tool. We'll open that up in here in just a second. And so with this prediction tool, you can see that we have now created a yellow dashed line that is predicting the yield value. And as we open this up, it's a multivariate prediction model where we have the yield value as the value we're modeling based on all these input engineering principles. We can choose the type of model, the regression method, and see kind of the predictions as to what the piece values are for how significant the variables are, or what the R-squared value is for the overall model fit. Now you can see that this model obviously isn't perfect, but it does give you an aspect of which yield values are higher or lower. So let's say that maybe this is good enough model fit for applying it online. So let's go and actually deploy this model. So you can see here that as this loads, there's a batch currently running. So right now we have a batch actively running, and you can see that by this hollowed out capsule at the top of the screen, which indicates that it's currently in progress. And so what we want to do is take that yield prediction and run it online right now. And so we've created this online monitoring range capsule to tell it when to deploy this model. And so all we're going to do is change that time range to current time and into the future. And you can see now we have a yellow dashed line there telling us what the current value of the yield is predicted to be based on the current aspects of what has gone into the batch so far. And so this is a really quick way to see, what's happening in the process and seeing this actually updating in real time as you're going along. Operators can use this information to actually affect, what's the batch going on, what's happening there, and how are things being deployed. At this point, I'd like to stop for a second and open it up for any questions. Elise, would you like to see if there's any questions put in?
We do have a couple questions right now. And again, you can submit your questions to the questions section in the GoToWebinar console. So the first question, what regression methods are available in Seeq?
Yeah, absolutely. I can take that one. So inside the prediction tool, we have 3 regression methods available. So there's ordinary least squares, ridge regression, and principal component analysis. However, if you want additional regression methods beyond that, that's where Seeq Data Lab can come in, because there's plenty of different options out there in the open source Python, R, or Octave environment that you can run in Jupyter Notebooks in Seeq Data Lab. And so if you want a partial least squares, OPLS, OPLSDA model, etc., those are all available in that aspect.
Great. The next question, I manage many CIP systems across sites. Would I have to recreate this analysis for each system?
That's a great question. So one thing that's cool about Seeq is that we leverage asset trees in such a way to duplicate analyses really quickly. So connecting to an asset framework or asset hierarchy where you have these different CIP systems inside an asset tree, we can really quickly use swapping functionality or tree maps to duplicate this analysis many, many times for each system across the site.
Excellent. Thank you, Joe. And that is all the questions we have right now, but we'll have another area opportunity for questions at the end of the webinar.
Okay, sounds great. So continuing on with the next example here, we have what we often call golden profile or golden batches. And in Seeq, this is called a reference profile. And so we're going to see how we can monitor quality, not just from the prediction tool as we did in the last example, but actually from a univariate modeling where we track the trajectory of batches over time based on historical batches. And so in this case, we're going to look at a fermentation process actually in the beverage industry. And in this fermentation process, there's an ending temperature profile that actually really impacts your product quality. And unfortunately, this quality data is actually measured offline, so it's critical to actually monitor these online parameters to ensure they're inside a particular range so that we don't impact that product quality as we finish out the fermentation. And so what we're going to do is monitor that temperature based on those historical curves, create that golden batch around each time that final operation occurs. And in this case, it's a diacetyl rest phase. And then apply that profile to future batches so that we can predict kind of like what that product quality and verify that it's within these bounds of what would be acceptable product quality. And so ultimately what we're going to try and do is improve that product quality by keeping that within a particular range. We're going to detect any early deviations or things that might be occurring that are incorrect for the process. We're going to optimize those production times to kind of stay at a consistent level. And so golden profiles in general are highly important for monitoring, and they can really be based on any sort of number of historical batches or profiles of interest. And these profiles can be, individual batches or the entire batch, or it can be as granular down to a individual unit operation or phase. And these profiles are often attached to, product codes where you make an individual profile for each product, or something like an MES system if you want to actually go down to every single phase within each product. So let's kind of see this in action. So what we're going to start off with is these fermentation batches. And so we're going to grab these fermentation batches with the diacetyl rest phase at the end of the batch, which is what we're going to focus on here. So you can see the diacetyl rest phase in the light blue, along with the online variable of temperature in the top lane and the offline variable of diacetyl level that is grabbed in the bottom lane. And so with this, what we really are looking at is let's first determine which batches are good. And so what we ultimately want is this diacetyl level to go below 0.1 ppm. So we're going to quickly set that limit of the yellow line there and identify the batches that actually meet that criteria at the end of the system. And so you can see with the cursors that the first two batches on the left did not meet the criteria, but the ending three did. What we're then going to do is use our reference profile tool to create those statistical boundaries. So the average, the +3 sigma and -3 sigma limits of those good batches. And you can kind of see this profile as we overlay all these different batches together. So you can see that some met the criteria and some of the bad ones did not meet that criteria. We can then apply this to more recent batches where we might not have quality parameters out yet. And we can apply these as scorecard statistical boundaries where it highlights them in red as to where we might not want to be. And then you can shift this sort of graph, this golden profile view over to Organizer where you might line it up with the scorecard, which tells you what the current temperature limit is and flags it red if it goes outside the limits and kind of shows you those profiles over time to quickly see where you might be having issues or not. We also want to illustrate here, what else can you do inside of Organizer topics? So we've shown kind of some quick use cases, but let's see like kind of the breadth of what you can do inside Organizer. So whether you want, boundaries that are moving and automatically updating graphs that tell you when there's deviations occurring, KPIs that are quantifying those deviations or particular statistics around CPPs that you might want, tree maps that are flagging what areas does a maintenance technician or an operator need to act on, whether it's a particular piece of equipment or a particular area of the site, or even notes that you just want to add in there to start documenting daily meeting sort of stand-up criteria that you might be looking at. Oops. The last use case today is going to touch on the drug product side of the business. And so in this use case, we're going to start with like the R&D scale and actually kind of act like we're developing out the drug product process. And we're going to focus on a twin screw granulation step. So in the process development of a drug, there's typically hundreds of experiments and batches that are run throughout that overall time. And so as you run these DOE experiments and things like that, we get a lot of insights and we get this process refinement that's going along. And so what we want to do is actually take that information and that knowledge that's captured at the R&D scale and actually apply it to the commercial scale. So as we go through a pilot scale and commercial production, we want to have those DOE limits actually in operation as we go along. So this allows you to kind of capture all that knowledge between R&D, pilot, manufacturing, and overlay the experimental data with batches that are running out the other side of the plant, along with utilizing that journal feature to give you those analytics, breadcrumbs, and reports and things like that that might be available. And so what we're going to do here is build a quality by design model. So it's a model that is based on multiple inputs and apply that model to the commercial data once it's developed. Quality by design, if you're not aware of it, it's a highly important subject for quality assurance and regulatory aspects, where they want to provide a process that actually ensures the high quality materials. And so in this use case, we're going to describe how to utilize and document that quality by design approach so that we can technically transfer that across from the R&D data that's built on a DOE across to the manufacturing to try and minimize those quality deviations and flag those deviations as they happen in near real time. So let's kind of see this in action here. So in this process, we have an API that's being added with excipients and water to a twin screw. And at the outlet of the extruder, we're seeing quality measurements such as granule size distribution being analyzed. And so when we think about this process, kind of the first step we want to do is actually gather that data. So in this case, you can see on the screen, there's 17 disparate experiments from that DOE. We've got the data just going offline in between. We want to use that to build our model. We can add all that data to the screen with the quality data that we've collected and kind of use that to build our model. So in order to set a design space, we want to generate a multivariate model of the quality data based on that process data. However, there are a couple tricks to creating an effective model here. So for a given particle moving through the process data and quality data are taken at different times along that twin screw. Using Seeq, we can align that data using the delay operator so that we can actually see when a material is passing through rather than just taking a single point in time. In addition, we want to limit this data to just a steady state period of time. So rather than getting the data that's starting up and shutting down, we want to limit that model to just those capsules that are identified on the screen there, which is that time when the process was able to get to steady state there. Now, once we have this data isolated, we can actually go and develop this model. And so in that prediction tool that we've shown before, we can create those multivariate models. So in this case, we created models for the fine counts, the percentage of fines, and the average diameter. And you can see the drug product specifications over there in the journal. And so what we wanted to do was create which parameters going into that would affect those quality parameters. And so in this case, we used screw speed, throughput, and liquid to solid ratio, which are all calculated variables, into this model. And you can see that the screw speed was actually not significant in this model. The p-value was greater than 0.05. And so we can remove that and just use a two-component model if we wanted to get the quality parameter based on that yield, or based on those liquid to solid ratio and throughput values. And so using these statistical models, we can actually, invert the model and calculate out what the entire design space would be. And so we can apply this as a multivariate function where we've got the quality parameter being calculated based on those two model input parameters, which give you this aspect of a moving boundary over time because it's being calculated based on another moving variable. And so you can see here the two input variables at the top of the screen and the two quality parameters at the bottom of the screen. And the capsules being shown that tell you at the top of the screen, when is this parameter out of its particular window that you want to operate. And you can set this to auto update and have it in near real time. As new data comes in, we can continue to get that deviations flagged and identified immediately. And with that, that was the end of the use cases today. Are there any more questions, Elise?
So we've had several questions come in, and some of these may be a little more complicated, and we can follow up with you following the webinar with a more detailed explanation. But the first question, we have cell cultures across multiple bioreactors. Can we create a golden profile across multiple reactors?
Yeah, absolutely. So we have many examples where you might have like a seed train where you've done a cell culture crossing across many different bioreactors. And so we actually have an example of this up on our Seeq.org community forum that goes through exactly how to do this. It's a little complicated to cover today, so I'll just kind of refer you to that aspect of it's available out there on the open web if you just go to Seeq.org.
Okay, the next question. Can you apply a golden profile from lab data to pilot or commercial data?
Yes, again, this is absolutely possible. So what we often see is that people develop those lab data sets, and they've got that center point process that they're actually going to run in the pilot or commercial, and they can just really simply apply it to a different condition inside our reference profile tool and allow you to apply it to a particular signal that might be different than what you're making the golden profile off of.
Great. Bryon, I think this question is for you. What is one of the more complicated issues that Grantek has seen solved through the use of analytics?
Sure, yeah, I can answer that one. So, I spent a few years as an embedded engineer in a biopharmaceutical contract development manufacturing organization, and their biggest concern was scheduling the multiple products and the multiple customers that they were manufacturing for. They had on the critical utilities and on the support equipment side, they had one purified water loop, one hot WFI loop, a couple ambient WFI loops, one area for buffer manufacturing and storage, and then 5 different CIP systems. So they're constantly, looking for ways to optimize. Well, number one is buffer manufacturing, and the other was CIPs, so they could schedule more batches. Cleaning in particular was a major bottleneck, for this company. So, and keep in mind this was quite a few years ago. So I was limited to using Excel and some basic trends to analyze their CIP cycles, but still we're able to drive a few optimizations around, for example, like how they were adding chemicals to make up the cleaning solution, and we did find quite a few irregularities, such as there were a bunch of differences in cycle performance across these 5 supposedly identical CIP units, but the availability of a more sophisticated tool would have undoubtedly been beneficial to further debottleneck the cleaning process and and to get them able to squeeze more batches out of those multiple production lines.
Excellent, thank you. And the last question we'll get to during the session, can we apply new process knowledge other than what's configured in the system?
Yeah, absolutely. So there's different ways to get your information in there. So we obviously have a wide variety of different connectors to get to those different data sources. But ultimately, you can really add any sort of knowledge into there with the organizer and journal aspects. So being able to type in any sort of notes, copy and paste it in, or even provide links to other aspects of information and data that you might have.
Great. Thank you to both the presenters, Bryon and Joe. If you have any additional questions about Seeq, or if you'd like to schedule a live demo, please email us at info@grantek.com. As always, we encourage you to connect with us on social media and to visit our YouTube channel to access video tutorials. That concludes today's webinar. Thank you for attending.